तो आज हम अपना सेकंड पार्ट सेक्सुअल रिप्रोडक्शन प्लांट का सेकंड पार्ट रिकॉर्ड करने जा रहे हैं। ठीक है जी। चलो शुरू करते हैं। डबल फर्टिलाइजेशन। After entering one of the synergid, the pollen tube releases the two male gametes into the cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuses with it, thus completing the syngamy. This results in the formation of diploid cell, the zygote. The other male gamete moves towards the polar nuclei located in the central cell and fuse with them to produce a triploid primary endosperm nucleus (PEN). As this involves the fusion of three haploid nuclei, it is termed triple fusion. Since two types of fusion, syngamy and triple fusion, take place in an embryo sac, the phenomenon is termed double fertilization, an event unique to flowering plants. The central cell after triple fusion becomes the primary endosperm cell (PEC) and develops into endosperm, while the zygote develops into an embryo. Post fertilization structure and event. Following double fertilization event of endosperm and embryo, development mature of ovules into seed and ovary into fruit are collectively termed post-fertilization event. Endosperm. Endosperm developed precedes embryo development. Why? The primary endosperms divide repeatedly and forms a diploid. Endosperm Endosperm tissue. The cells of this tissue are filled with reserved food material and used for the nutrition of the developing embryo. In the most common type of endosperm development, the pen undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occur and the endosperm became cellular. The number of free nuclei formed from cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of cells, and the surrounding white kernel is the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, example pea, groundnut, beans, before seed maturation, or in it may persist in the mature seed, example castor and coconut, and to be used up during seed germination. Split open some seed of castor, pea, bean, groundnut, fruit of coconut, and look for the endosperm in each case. Find out whether the endosperm is persistent in cereals, wheat, rice, and maize. Embryo. Embryo developed at the micropylar end of the embryo sac where the zygote are situated. Most zygote divide only after a certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though the seed differed greatly, the early stage of embryo development, embryogeny, are similar in both monocotyledons and dicotyledons. Depict the stage of embryo in a dicotyledonous embryo. The zygote gives rise to the proembryo and subsequently to the globular, heart-shaped and mature embryo. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledon is the epicotyle, which terminates with plumule or stem tip. The cylindrical portion below the level of cotyledon is hypocotyle, that terminate at its lower end in the radicle or root tip. The root tip is covered with a root cap. Embryo of monocotyledon possess only one cotyledon, and the grass family. The cotyledon is called scutellum. That that is situated towards one side, lateral of it, of the embryonal axis, and its lower end, the embryonal axis has the radicle and root cap enclosed in an undifferentiated sheet called coleorhiza. The portion of embryonal axis above the level of attachment of scutellum is the epicotyle. Epicotyle has a shoot apex and a few leaf primordia enclosed in an hollow follicular structure, the coleoptile. Seed. In angiosperm, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Seed are formed inside fruit. A seed typically consists of seed coat, cotyledon, and an embryonal axis. The cotyledon of the embryo are simple structures, generally thick and swollen due to storage of food reserved as in legumes. Mature seed may be non-albuminous or ex-albuminous. Non-albuminous seeds have no residual endosperm as it is completely consumed during embryonal development. Example: pea groundnut. Albuminous seed retain a part of endosperm as it is not completely used up during embryonal development. Example: wheat, maize, barley, castor. Occasionally, in some seeds such as black pepper and beet, remnants of nucleus are also persistent. This residual persistent nucleus is the perisperm. Integument of ovule harder as tough protective seed coat. The micropyle remains as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. As the seed matures, its water content reduces and seed become relatively dry, 10 to 15% moisture biomass. The general metabolic activity of the embryo slows down. The embryo may enter a stage of inactivity called dormancy, and if favorable conditions are available, adequate moisture, oxygen, and suitable temperature, they germinate as ovules mature into seed. The ovary developed into a fruit. Example, that is the transformation of ovule into seed and ovary into fruit proceeds. Simultaneously, the wall of the ovary develops into the wall of fruit called pericarp. The fruit may be fleshy as in guava, orange, mango, etc., or may be dry as in groundnut and mustard, etc. Many fruits have enclosed mechanism of dispersal of seed. Recall the classification of fruit and their dispersal mechanism that you have studied in earlier class. Is there any relationship between number of ovules in an ovary and the number of seed present in a fruit? In most plant, by the time the fruit develops from the ovary, other floral parts degenerate and fall off. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contribute to fruit formation. Such fruit are called false fruit. Most fruit, however, developed into developed only from the ovary and are called true fruits. Although in most of species fruits are result of fertilization, there are there are a few species in which fruits developed without fertilization. Such fruit are called parthenocarpic fruit. Banana is one example. Parthenocarpy can be induced through the application of growth hormones, and such fruit are seedless. Seeds offer several advantages to angiosperms. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seed have better adaptive strategies for dispersal to new habitat and help the species to colonize in other areas. As they have sufficient food reserved, young seedlings are nourished until they are capable of photosynthesis and their own. The hard seed coat provides protection to the young embryo. Being product of sexual reproduction, the, they generate new genetic combination leading to variation. Seed is the basis Seed seed is the basis of our agriculture. Dehydration and dormancy of mature seed are crucial for storage of seed which can be used as food throughout the year and also to raise crop in the next season. Can you imagine agriculture in the absence of seed or in the presence of seed which, which germinate straight away soon after formation and cannot be stored? How long do the seed remain alive after they are dispersed? This period again varies greatly. In a few species, the seed lose viability within a few months. Seed of a large number of species have for several years. Some seed can be remain alive for hundreds of years. There are several records of very old yet viable seed. The oldest is that of a Lupinus arcticus, excavate from Arctic tundra. The seed germinate and flowered after an estimated record of 10,000 years of dormancy. A recent record of 2,000-year-old viable seed of the date palm Phoenix dacty, dactifera, discovered during the archaeological excavation at King's Herod Place near the Dead Sea. After completion, a brief account of sexual reproduction of flowering plants, it would be worth attempting to comprehend the enormous reproductive capacity of some flower plants by asking the following questions. How many eggs are present in an embryo sac? How many embryo sacs are present in an ovule? How many ovules are present in an ovary? How many ovaries are present in a typical flower? How many flowers are present in a tree? And so on. Can you think of some plant in which fruit contain very large number of seeds? Orchid fruit are one of such category, and each fruit contains thousands of tiny seeds. Similar in the case in fruit of some parasitic species such as orbanchi and striga. Have you seen a tiny seed of ficus? How large is the tree of ficus developed from that tiny seed? However, how many billions of seed does each ficus tree produce? Can you imagine any other example in which such a tiny structure can produce such a large biomass over the year? Epimixes and polyembryogeny. Although although seed in general are the production of fertilization, a few flowering plants such as some species of asteraceae and grasses have evolved a special mechanism to produce seed without fertilization called apomixis. What a fruit produce without fertilization called thus apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seed. In some species, the develop the diploid egg cell is formed without a reduction division and developed into the embryo without fertilization. More often is in many citrus and mango. Varieties. Some of the nuclear cells surrounding the embryo sac start dividing, protrude into the embryo sac, and develop into the embryos. In such species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryogeny. Hybrid varieties of several of our food and vegetable crops are being extensively cultivated. Cultivation of hybrids has tremend- tremendously increased productivity. One of the problem of hybrid is that seeds have to be produced every year. If the seed collected from hybrid are shown, the plant in and progeny will segregate and do not maintain hybrid characters. Production of hybrid seed is costly, and as the cost of hybrid seeds become too expensive for farmers. If these hybrids are made into apomictic, there is no segregation of character in an hybrid progeny. Then the farmer can produce Then the farmer can keep on using the hybrid seeds to raise new crop year after year, and he does not have to buy hybrid seed every year because of the importance of apomixis in hybrid seed industries. Active research is going on in many laboratories surround the world to understand the genetic of apomixis and to transfer apomictic genes into hybrid. तो यारो ये हुआ हमारा चैप्टर समाप्त. सेक्सुअल रिप्रोडक्शन इन फ्लावरिंग प्लांट चैप्टर हो चुका है हमारा समाप्त. वीडियो को देखने के लिए थैंक यू।